Hello everyone and welcome to Instant Biology by Dr. Neela. I am here with you again with a very interesting topic and the topic that I would be dealing today is lambda phage vectors. Now for those of you who understand Hindi better, I have already made a video on lambda phage vector in Hindi, and those of you who are comfortable in English can carry on with this particular video. Now, I would be giving the link to the Hindi video at the end of this particular video at the top. You can check it out. Also, let me just brief you what all we would be dealing with in this particular lecture before going into the depth. So I would be talking about the structural features of lambda phage vector. I would be talking about the working of the lambda phage vector, how gene of interest is inserted inside the lambda phage vector, what or how the insertional lambda phage vector is created, how the replacement vector is created. Apart from that, I would also be talking about some of the previous year questions that have been asked on this topic. So all in all, you would have a complete understanding about this vector if you go through the video up till the last. So without much ado, let me start with the topic. So the first thing that you can see, or the first point that you can see on your screen, is size of lambda genome. Now you would be knowing that lambda phage. Firstly, let me make you understand what are lambda phages, or if I most simply say what is a phage. So you would be knowing that phage or phages, these are the viruses, and these viruses have the capacity to infect bacteria or treat bacteria as hosts. That is why these viruses are called as bacteriophages. They are also referred to as bacteriophages. Now what happens is, when a virus infects a bacteria, see, for example, let me just show you. So suppose this is a bacteria, and let me show you that this is a virus. Let us say this is a bacteriophage virus, right? So it has its genome over here, and when this virus will be infecting the bacteria, what will happen? The genome of the virus will move inside the bacteria, and it will gain control of the bacterial cell, and it will command the bacterial cell to do whatever it wants. So this is how a phage vector performs its function. We would be discussing more about the phages as we move along in this particular lecture, but for now, let me just give you an idea that this lambda phage genome or lambda phage vector is having a genome. So all the viruses would be having the genome. So this lambda phage vector also has its genome. We are calling it as lambda genome. The size of the lambda genome is 48502 base pair. So if I convert it into kilobase, so it would be roughly around 48.5 kb. So this is the size of the lambda genome. Now, what all are the genes that are present inside the lambda genome? This is also important because the genome, I have given you the size of the genome, but you should be knowing that what are the genes that are present inside this genome. For example, the gene of origin of replication is present. Now, what do you mean by origin of replication? This particular portion will be responsible for replication inside the host. That means if my lambda phage affects or infects a particular bacteria, the genome can replicate inside the host or E. coli. So for that purpose, this origin of replication gene is present. So this is important. Next is genes for head and tail protein. I would be showing you this with the help of a diagram, that what is a head and what is a tail, and the proteins that are required for synthesizing the head and the tail. We would be going through that also. Apart from that, DNA replication, the genes that are required for DNA replication, they are also present inside the genome of the lambda phage. Obviously, when the vector or when the genome of the lambda phage enters inside the bacteria, it has to undergo replication, it has to make its copy. Therefore, DNA replication genes are present. Genes for lysis and lysogeny are also present. Now, what do you mean by lysis and lysogeny? Let me explain this to you with the help of the diagrammatic representation. Now, in the previous, when I was explaining to you what are bacteriophages, I told you that the bacteriophage vector has a genome, and when this vector enters or when this vector infects the bacteria, the genome of the bacteriophage enters inside the bacteria and takes control of the entire bacterial cell. Now, it can have or it can process or it can undergo two cycles. The first cycle is lytic cycle. Let me first of all tell you about what is lytic cycle. In case of lytic cycle, the entire genome of the entire genome of the virus enters inside the bacteria. It takes control of the entire bacterial cell and it makes copies inside the bacterial cell. That means it replicates inside the bacterial cell utilizing the bacterial resources, and after some time it will create lysis inside the bacteria. That means bacteria will be dying and all the viruses that are produced inside the bacteria will be coming out. This is called as lytic cycle. Now the next cycle or the different type of cycle that can be executed is called as lysogenic cycle. What do you mean by lysogenic cycle? In this case, everything is same. That means the vector, that means the virus will be coming, it will be infecting the E. coli, the genome of the virus will be coming inside the bacteria. But now over here, the virus will not, or the viral genome or the virus will not cause lysis inside the bacteria. Then what will happen? The genome of the virus will be integrating inside the genome of the bacteria. What do I mean? So something like this. So suppose this was the bacterial genome. This is the genome of the bacteria. Now the genome of the virus, something like this. So suppose this was the genome of the virus. So genome of the virus will be integrating inside the genome of the bacteria, and this can be continued for years together. That means in this case, the virus will not be causing lysis inside the bacteria and it will be producing its copies just like that. So the bacteria will not be killed. But one fine day, because of some kind of stress situations or because of some kind of trigger, the lysogenic cycle can be converted back into the lytic cycle, and again the bacteria can lyse. So this process is called as, this cycle is called as lysogenic cycle. I've already told you about the lytic cycle. Now there are specific genes inside the virus that trigger lytic cycle or can trigger the lysogenic cycle. So that is what I was saying, that so DNA replication genes, genes responsible for lysis and lysogenic cycle. Apart from that, single stranded protruding ends of 12 bases are also present. Now, what do I mean when I say single-stranded protruding ends? I want to say that these ends will not be base paired with the other strand. For example, what I want you to understand is, so something like this. In this case, you can see that this end over here is protruding and this end over here is protruding. That means overhangs. We can say that these are overhangs. So in case of the lambda phage vector, the genome has overhangs and these overhangs are of 12 bases. So this is the configuration of the bases that are the configuration of the overhang that is present. Okay, so it is present on both the ends. So for example, it is present over here also and it is present over here also. I would be talking more about this as the lecture proceeds. So now let us move on to the next slide. Okay, so as I've already told you that what are the genes that are present inside the lambda phage genome, but here is a structural representation which would give you a better understanding about the genes. So first thing that I want you to focus on is that this is the structure of lambda phage, right? So this is the portion of the head. So this is the lambda phage and this is the head portion. So this is the head portion, and inside the head the genome is present. So let us say that this is the genome, this is the collar portion of the phage, this is the tail portion that I told you, and these are tail fibers which are used for attachment on the host. Okay, so this is also the base plate. So this is the structure of the lambda phage. Now coming to the genome of the lambda phage, understand that the genome is present inside the head region of the lambda. Okay, so this is the genome. Understand that when the genome, if you are talking about the genome that is present inside the head of the lambda phage, it is present in the linear form. As you can see over here, this is the linear form. So it is present in the linear form when it is present inside the lambda phage. That means it is present inside the virus. But when it goes inside the host, when it moves inside the host, then what happens is it gets circularized. This is an important thing. Now you can see over here, these are the genes that are present for the purpose of DNA replication, the genes for lysis, the genes related to head formation, tail formation, ATT region. So these are some of the genes that are present. I have already told you about them. So that is enough. Now let us move on to the next slide. Now, as I've already said that the genome remains linear in the phage head. So in this particular region, when it is in this lambda phage form, let me draw like this. So in the head region, it is present in the linear form. But as it gets transferred inside the E. coli, or when I say E. coli, I mean the host. So inside the host, it gets circularized. Okay. Next. Sealed cohesive ends are also called as cos sites. I want you to focus upon these regions. Now, this region over here and this region over here. These are called as cos sites. Now, I've already told you that 12 base pair regions are present as overhangs. So we are talking about these regions only. The overhangs are actually the cos sites. Okay, so these are the cos sites. They are not paired. They are unpaired regions that are extending, and they are not paired 12 base pair regions on both the ends. So that is why I've written over here is sealed cohesive ends are called as cos sites. Now when the genome enters inside the bacteria or the E. coli or the host, what happens is these cos sites will come one over the other, and in this way the packaging, or in this way the circularization of the genome would become easier. So the purpose of cos site is to help in circularization. Now when the genome wants to again become linear, the cos sites would be cut off and it will again become linear. So sealed cohesive ends are called as cos sites. Over here you can see that the cos sites are over here sealed. That means they are again paired. So the cos site over here pairs with the cos site over here. So it becomes circular. Again, when it wants to become linear, the cos sites are cut and they become linear. So this, I believe you would have got a clear understanding. Cos sites are sites of the cleavage. I have already told you this. Also play a crucial role in packaging of the mature phage DNA into phage heads. This is again an important point. Most of the questions are asked on this point only, that what is the purpose of cos sites. Primarily cos sites are used for the circularization. This is one thing. Again, another thing is cos sites are also utilized for packaging the genome, the lambda genome, inside the phage head. So when I am packaging this inside the phage head, so this, for this purpose also cos sites are used. How they are used, we would be talking about that a little later. Now this is what I was talking about. So this is a diagrammatic representation of exactly what I was telling. So this was a cos site over here, this is a cos site over here. Both the cos sites are paired now, and they come close to each other. They are complementary in sequence and they get paired. In this way the circular DNA is formed. Right, now let me move on to the next slide. Two problems in using lambda phage as a vector. Now in general, the lambda phage vector is a very nice, is a very a usable kind of a vector. But the problem is, there are two problems majorly when you are using lambda phage vector. The first problem is related to the size. As you already know that the size of the lambda DNA should be between 38 to 52 kb. Now I've already told you roughly it is around 48.5 kb. It can be extended up to 52 kb. The general average is roughly around 48.5 kb. This we know. So the lambda DNA size should be between 38 to 52. We are keeping it as 48.5 kb. Therefore, the lambda genome can accommodate 3 kb of DNA insert only. What I want you to understand is that generally, or on an average, 48.5 kb is the size of the lambda genome. It leaves you with only 3 kb window of your gene of interest to be inserted. Now, why are we talking about the lambda phage over here? Because we want to use the lambda phage as a vector for transferring our gene of interest inside the E. coli. Now, how would you transfer your gene of interest inside the E. coli? You will have to put your gene of interest inside the vector, and automatically, since this is a phage, it has the capacity to infect the bacteria and to transfer its own DNA inside the bacteria. Now, when it has the capacity to transfer its DNA, automatically your DNA would also be transferred inside the bacteria, because your DNA or your gene of interest is integrated inside the viral genome, right? That is why we are interested in this. But the problem is that the size of the genome is very large for lambda phage vector. It is 48.5 kb, which leaves us with only roughly 3 kb of the window that we can insert our gene of interest, because maximum is 52 kb. This is the maximum size. When I say maximum size, why am I saying maximum size? Because I have already told you that the genome would be integrated inside the head. Genome has to go inside the head. So the capacity, the maximum capacity of the head itself is 52 kb. So you cannot exceed that limit. Therefore, this leaves us with only 3 kb of DNA insert window. So this is one problem. Now, every scientist who is working on a lambda phage vector would want to increase this length, because I would want to transfer more of my gene of interest, because that is essential for me, right? So what is the resolution of this problem? The resolution is to delete the central region of the lambda genome. Now, let me give you an example. Suppose you are living in a room. The room is already preoccupied with a lot of stuff. There is a lot of stuff. There are a lot of things that you are not using. Now you can bring new things only when you are eliminating or deleting out or putting or discarding the older things that were present. So the same thing has to be applied over here. The lambda genome itself is very big in size. You can put your gene of interest only when you are deleting some of the portion of the lambda genome. So that is what we are saying over here. Delete the central region of the lambda genome. So what is the central region? This is a region that is not very essential for our purposes. Of course, it is essential for the lambda vector, lambda phage, but it is not essential for our purposes. So if we delete out this particular central region and then put our gene of interest in place of this, then we would be getting a larger window. That means 20 to 35 kb of region is or can be deleted. The central region is roughly around 20 to 35 kb. This is non-essential for us, but it is essential for the virus to cause lysogenic infection. Lysogenic cycle. I've already told you that virus can be going through lytic cycle or lysogenic cycle. Suppose we are removing this, then the virus will not be able to undergo the lysogenic cycle. We do not care about that. So this region can be deleted, and in place of this region we can put our gene of interest. So if you do this, there will be no effect on the ability of a phage to infect E. coli cells. Definitely, this is always kept in mind, that whatever modifications we are doing inside the virus genome, it does not hinder its ability to cause infection inside the E. coli, because that is the primary step that has to be taken up, or that has to go along. Only then our gene of interest will be integrated inside, or will be reaching inside the bacteria. I hope you got a clear understanding about this. So if we follow this process, what we will be getting, we can get larger DNA inserts. That means our gene of interest, a greater size or a bigger size of our gene of interest can be put inside the bacteria. And the second thing is, recombinant phage will always be lytic in nature, because you have deleted out the lysogenic region that was responsible for performing the lysogenic cycle. You have deleted out that. So obviously the only option left for the vector is to undergo the lytic cycle. I hope you got this clear. Now, let me show you with the help of a diagrammatic representation of what I am aiming to make you understand. So this is the lambda phage vector. Now this is the lambda DNA molecule. So this is one end and this is second end. So these are the two ends. and in between these two, central gene cluster or the central region is present. I've already told you that central region is responsible for the lysogenic cycle. What is being done over here? The central region has been removed by enzyme digestion. Central region is removed. Now what is done is our region of interest or our gene of interest, this is integrated in between the first and the second region. I mean to say is left and right arm, we can say that, not the first and second region, but left arm and right arm. So between the left arm and right arm, your gene of interest is integrated. In this way, we can integrate larger size of the gene of interest. This is an important thing that you must understand. So this is the concept. Now, let me move on to the second problem. I already told you that there are two problems associated. The first problem was the size. Now, what is the second problem? Second problem is, as I've already said, that the lambda genome size is very large. Also, that inside the lambda genome, there are many restriction sites for one restriction enzyme. Let us understand this again. So, in general, what happens is one restriction enzyme has one restriction site allocated to it. That is why, suppose, let me show it to you. Suppose this is a DNA fragment. It has a site over here, and this is eco R1 site. So when I cut it with eco R1, what will happen? This kind of a structure will be present because eco R1 cuts in this particular region. But in case of lambda genome, the genome is very big and it is having multiple sites for one restriction enzyme. For example, for BAM-H1 it is having 2 sites, for EcoR1 it is having 3 sites, for Hnd3 it is having 2 sites. Now because of this, what happens is, whenever you are cleaving with any enzyme, for example, if you are cleaving with EcoR1 and there were 3 sites of EcoR1, the vector itself will be fragmentized. Now this is not a very good thing to happen to a vector. The vector itself will be breaking down into number of fragments. This is not a good thing to happen. So in order to resolve this problem, what is done is we form mutant lambda genome. That means some kind of mutation, some kind of modification, some kind of aberration is done inside the lambda genome so that it now does not have the restriction site, or it lacks the restriction site. Now it has only one or two restriction sites for roughly 1 or 2 enzymes. That means one or two restriction sites or 1 or 2 enzymes only can cleave at that cleave the genome. So multiple restriction sites concept is now removed. Why? How? Through creating mutations or through creating aberrations. I believe you would have got a clear understanding about this. Now, so as I've already said, you have to perform mutations and recombinations in the lambda vector in order to make them improved or in order to improve their efficiency. So the mutation and after the mutation and recombination, 2 features are seen. One is the lambda phage vector can be propagated inside the E. coli cells as phage. That means inside the E. coli cell, the phage genome, it gets replicated, and concatamer formation is also seen. Now what do we understand by concatamer formation? Now in order to understand this, let me show it to you with the help of a diagram. Suppose this is a lambda genome. Okay, this is a lambda genome. Now this is the non-essential segment. As I have already told you about the non-essential segment, what it is, we can delete this. And 2 eco R1 regions are present. So this is one eco R1, this is another eco R1. So we can cut it out. So when we cut, so this non-essential region has been removed and it has to be discarded. Now we are left with left arm and right arm. Now we are having eukaryotic DNA, or you can also call it as gene of interest. So this gene of interest is integrated between the left arm and the right arm like this. Right? So this is called as the recombinant DNA. Why are you calling it as a recombinant DNA? Because let us say this is the lambda phage DNA, and you are integrating your DNA of interest over here, and then something like this has been done. That means these two are the lambda genome, and this is your gene of interest that you have integrated. That is why it is called as recombinant DNA. Recombinant DNA. Now After forming the recombinant DNA, still the cos sites are present. Now, with the help of these cos sites, another vector, another lambda genome will be integrating with the previous one by utilizing the cos site. So you can see over here. So this is one genome, and it is integrating with the second genome with the help of the cos sites. This is called as concatamer formation. Concatamer means the extension or the joining of the repeats. The joining of the repeats over here. The repeat is this one. So this is one repeat, and it is joined by another repeat with the help of cos sites. So this is called as concatameric formation. After the mutation and after the recombination, this property can be integrated. Now let us understand about the classification of the lambda vector into two groups. Now for ease of understanding, I can classify it into two groups. The first one is insertional vector and the second one is replacement vector. Now, in case of insertional vector, what happens is, I have already told you that the some of the non-essential regions are present inside the lambda genome. You can cut out the non-essential regions. So non-essential region has been removed. The right arm and the left arm are left. They are joined together. Right arm and left arm are joined together. Now after this, what happens is, in different types of lambda genomes, you use different genes. Now different genes will be present in different lambda genomes. Now in between these genes, you can integrate your gene of interest. For example, let me explain it to you. So for example, in case of lambda GT10, this is a kind of a vector. In lambda GT10, there is a gene called as C1 gene. Now in this C1 gene, you can integrate you can insert your gene of interest. Similarly, in case of lambda GT11 or lambda ZAP2, in this kind of vectors, you can insert your gene of interest inside the lac gene, basically, right? So this was about the insertional vector. Let me talk about the replacement vectors. What do you mean by replacement vectors? In case of replacement vectors, deletion of all or major part of non-essential region of lambda genome is done. So all, complete, or major part of the lambda genome is major part of the non-essential region of lambda genome is removed out. Now the deleted region or the region that has been removed is called as a stuffer fragment. Only and only two restriction enzymes or sites are present, or everything else is removed. Now can be used to clone much larger DNA inserts. Obviously, if you are removing so much of a region, then the window for integrating your gene of interest becomes very big. So roughly lambda EMBL4, that is if I just move out of the way so that you can see, lambda EMBL4 is kind of a vector, and that has a 20 kb insert size. So this is again a very interesting thing. So roughly, what is the insert size that you can integrate? Roughly 15 to 20 kb, roughly around that you can integrate inside a lambda genome because of the modifications that can be done. Now let us move on to, obviously replacement vectors would have more of a window as compared to insertional vectors. Now let us move on to the next thing. Let us talk about the PYQs on lambda phage vectors, right? So first question that comes up is, this has been asked in GATE BT 2015. Which of the following statements about lambda phage vector is true? Lambda phage vector can clone DNA fragments larger than 50 kb. No, this is wrong. It's roughly around 15 to 20 kb max. Plasmid vectors based on lambda phage can replicate as phages in bacteria. Not exactly as phages. The cos sites are required for DNA packaging in the phage particle. Exactly, this is true. Lambda phage vectors cannot be used. It can be used for genomic library construction because of the big gene insert size that can be used over here. So correct option is C over here. Let us move on to the next question. So which of the following DNA inserts can be cloned using a lambda phage vector? Basically, lambda phage vector can have an insert size of roughly 15 kb that I have just told you, just gave you an idea about that. So these were the two questions that I thought would be essential for giving you a proper understanding. I believe you would have understood this particular lecture pretty nicely. If you have understood, give give a like, push the like button. Do not forget to subscribe the channel. Also share with your friends so that we can reach a wider audience. And also do not forget to comment in the comment section that which topics you want to study next. Thank you and have a good day.